Okay, so in this little video, I'm going to go over what we've just done with karyotyping, and what happens when things don't go quite according to plan. So just a quick recap, we have 23 pairs of chromosomes. One of each pair comes from mum, one of each pair comes from dad. In all people, whether you're male or female, the first 22 are what we call matched pairs. That means they not only are they the same size and same shape, they carry information about the same characteristics. So you may on this one here have characteristics for eye color from mum. On this one here you have characteristics for eye color from dad. When you get to chromosome pair 23 is where we get a few slight differences, because females are what we call XX, and you can see that they are in fact a homologous pair of chromosomes. They're a matched pair, they carry information. You got one from mum, you got one from dad. Okay, just like all the other 22. However, in males, you can see you have the X and the Y. They are not a matched pair. And when we get on a little later and we start talking about sex-linked inheritance, characteristics that are inherited differently if you are a male or a female, such as something like red-green color blindness, those characteristics are carried on the chromosome pair number 23. Now, you have heard of Down syndrome, I'm sure. And Down syndrome is an abnormality that occurs with chromosome pair number 22. And it goes back in meiosis to what happens here when the chromosomes split and go into the 4 gametes that are produced. And a mistake gets made, and this is what you get. In chromosome number 21, you get something called trisomy 21. And if you can see here, tri means 3, so you see you got 1, 2, 3 chromosomes instead of 2. What we're going to look at is why that happens. And it could be any of the pairs of chromosomes. It could be 1 through 23. It just so happens that if it happens in 21, that is what a person who would do karyotyping, you remember we talked about how women have, with high-risk pregnancies, get offered amniocentesis, where they can check the chromosomes that come from the baby and to make sure everything is normal, or to screen for certain problems. That would get picked up in here. There's also another one that, and there are lots and lots and lots of these, all right? But I thought I'd talk about this one, since this has actually been an issue in the international sports scene, and that's Klinefelter syndrome. And you can see here we have an X, we have an X, and we have a Y. So we have 1, 2, 3 chromosomes. So this would also be trisomy because there's 3 of them, and it would lead to, instead of having just 2 X's, but you've got an X and X and Y. And we will discuss that in a little more detail in class. I want to talk about how do we end up with 3 chromosomes. And for that, you're going to need this sheet. And I've shown you how to color your chromosomes. I'm going to cover it up because I want to go through things step by step so that you actually understand what's going on. So the first thing that happens here we have, and I'm only showing 1 chromosome pair because I'm not going to go draw 46 of them. It would get way too complicated. So the first thing that happens, you notice that these chromosomes now have traded out bits. So what did we have going on over here? At this point here, we must have had some crossing over, because crossing over is when the chromosomes trade out parts of their genetic material, their DNA, and that leads, as you remember, to genetic diversity. That explains why we are similar to our siblings, but we are not identical. And at that point, we can go through all the phases. We're not going to go through all the phases of meiosis, but you can see this one's going to go one way, this one's going to go this way. And what you end up with, I can do this, is these two cells here. You can see this chromosome has gone into this cell. And I'm going to say, for the purpose of this exercise, these are going to be egg cells. This chromosome has gone into this egg cell. Absolutely normal. That was anaphase, where they separated and they ended up in two separate cells ultimately. Now, if we look at the other one, we had a slight oops, because these chromosomes probably attached to the one of them attached to the wrong spindle fiber. So during anaphase, when the chromosomes were supposed to separate out, what happened is instead of going in opposite directions, you can see they've both gone in the same direction. So both of these chromosomes here have ended up in this cell, and none of this chromosome pair has ended up in this one. So you can see these are eggs because we started with one cell. We've got 1, 2, 3, 4. Okay, and this is normal. This is normal. This has 2 chromosomes in it rather than 1, which is abnormal, and this has no chromosomes in it, or of this pair in particular pair, and that is also abnormal. So what would happen if, since these are egg cells, they all want to meet a sperm cell, correct? Because that is the one job of an egg cell. So if this one here or this one here, if this sperm cell, see it's got a little tail, and this egg cell, so either cell 1 or cell 2, one cell, one chromosome, the zygote, which would be this one, this would be the zygote, the fertilized egg cell, okay, it would be normal, okay? But what if a normal sperm cell fertilized this egg cell? Look what we get now. So this is the one we're talking about. Here's my sperm cell. It fertilizes this egg cell. What do we end up with? We end up with 1, 2, 3 chromosomes in, I need to cover that bit in there, 3 chromosomes in this zygote. So again, this one's normal because we're supposed to have chromosomes in pairs. This one has a abnormality because instead of them being in a pair, 2 of them, this one there in 3, this is what we call trisomy, okay? Because there's 3 of them. Now, what happens if a sperm cell fertilizes a egg cell like number 4 here that's got nothing in it? Now, what do we end up with? That little sperm cell, let's just move that around, this little sperm cell here is the only one providing chromosomes for this pair, and so instead of having 2, we only have 1, and this is called monosomy. Okay, mono means 1. Okay, so again, these are the zygotes. Only the top one would be considered normal. So again, why does that happen? Why do we end up with just 3 or 1? Well, it's because of what happens up here. Okay, this little deal here, this is called Nondisjunction. Okay, if you break the word down, it actually means what it says. Non means not, this means to break, and junction. So instead of them separating and going in opposite directions, right, here what this actually means is that both chromosomes end up in the same gamete, the same sex cell. Okay, so because they both move in, you can see my little arrows, they're both moving the same way, they end up in the same gamete, which means that one of them, one gamete out of the four, has no chromosomes from this pair representing in it. And the reason that I did this with eggs is that typically it's more likely to happen with egg cells, the female gametes, than it is with the male gametes. It is also more likely to happen, the older a woman is when she has the baby, the greater the probability of non-disjunction, this word here, non-disjunction, happening. But you've also got to think about it. If it's more likely to happen in females, in egg cells, then you've still got to have that, this egg cell here or this egg cell here still has to be the one that gets fertilized. And if you think about it, 50% of the eggs out of this meiotic cell division, the production of gametes, are perfectly normal. So there's a 50% chance of getting an abnormality, right, within the egg. And since there are way fewer eggs, that's why you get an increasing chance when you the older you get. But just to recap, non-disjunction is when the chromosomes, everything goes normal during meiosis until the chromosomes are supposed to separate during anaphase, and instead of them going in separate directions, both of them in a pair will go in the same direction. If this one with two chromosomes then gets fertilized, you end up with a zygote, a fertilized egg cell that's got 1, 2, 3 chromosomes in it instead of a specific pair. So if this pair up here was chromosome pair 21, and it was this egg cell that got fertilized, this would be trisomy 21. That's what Down syndrome is. But if it was this one that has no chromosome for 21 in it, you would end up with monosomy. Okay, so that's non-disjunction. The next thing I want you to do is to look at these karyotypes. They look slightly crazy, and I want you to figure out, where is the chromosomal abnormality? Is it normal? Is it trisomy? Is it monosomy? And at which pair of chromosomes? Is it 1, 2, 3, all the way through to 23?